Thank you, Dr. Ketan. And let us start the first session of today's scientific forum. I invite Dr. Yogesh A. Kulkani, who is an Associate Professor and Associate Dean at Shobhaban Pratapbhai Patel School of Pharmacy and Technology Management, SPKMs and NMIMs, Mumbai, India. He is an eminent academician and researcher. Over past 18 years, he has conducted numerous research projects in various fields, including herbal drugs, natural products, diabetes, diabetic complications, and neurodegenerative diseases. He has 110 publications in peer-reviewed journals to his credit and has authored many books. Dr. Kulkarni is recipient of prestigious DST Inspire Faculty Award, awarded by Government of India. He has been featured in top 2% scientists in the world, list published by the Stanford University and elsewhere in 2021 and 2022. Dr. Yogesh Kulkarni would share his interesting insights on role of Rasashastra in diabetic complications and evidence-based revalidation. Over to you, sir. And we also invite Dr. Mukesh Chowda to join the head table.
Dignitaries on the dais, off the dais, doctors, healthcare professionals, students, ladies and gentlemen. Good afternoon to everyone. So today I'll be talking on a topic exploring potential of Vasanta Kusumaka Rasa in diabetic complications, molecular pharmacology approach. So diabetes, everyone knows it is nothing but what? It's a chronic disease which occurs when the pancreas is no longer able to make insulin due to some defects in pancreatic beta cells or destruction of pancreatic beta cells. Not being able to produce insulin or use it effectively leads to increased glucose levels in blood, which we call it as hyperglycemia. And long-term high glucose levels, persistent hyperglycemia, is associated with different types of complication. We call them as diabetic complication. And everyone knows the prevalence of diabetes is continuously increasing, and we can say that in 2050, around 60 to 70% people, they will be suffering from diabetes. Now coming to diabetic complications, this persistent hyperglycemia damages important organs in our body if it is not well managed. Like it can affect eye, it can affect heart, it can affect kidney, neurons, brain, etc. So based on these damages, different types of organs which are being damaged, these complications can be categorized into microvascular and macrovascular complication. So when it is a small blood vessel which is responsible for damage to any specific organ, we call it as microvascular diabetic complication. And when large blood vessels are involved, we call it as macrovascular complications. So it leads to diabetic retinopathy, diabetic nephropathy, diabetic neuropathy, cardiomyopathy, encephalopathy, etc. So this persistent hyperglycemia management is very important aspect when we want to treat or manage diabetic condition. Now when we talk about diabetic complication, these diabetic complications, as their name suggests, they are too complicated when it comes to detailed pathophysiology. There are many factors like metabolic factors, uncontrolled glycemia, uncontrolled lipids, they are linked with each other. Different types of hemodynamic parameters, we generally see that diabetic people, they suffer from different types of cardiac disorders. Blood pressure, salt fluid balance, and of course the last one is the genetic factors, which can also play important role in development of these diabetic complications. Now all these factors, when we combine together, metabolic, hemodynamic and even genetic, it leads to different types of changes at cellular level. And these cellular changes, it may lead to modifications in gene or modulation of different types of proteins involved in progression of these diabetic complications. At the end, the immune cell recruitment at various organs leads to severe damage, causing cell dysfunction and cell death. This is applicable to all organs which face this persistent hyperglycemia. Vasanta Kusmakar Ras, it's a well-known medicine in Ayurveda. It's a classical Ayurvedic formulation indicated in Prameha, and it's a unique combination of minerals and herbs that provides natural immunity to body cells and help in the treatment of weakness which is caused in diabetic condition. Rasausidhis, they work on different slotas and penetrate into deepest tissues carrying highly potent charge. And of course, we know that VKR is an important formulation which is manufactured and marketed by various herbal drug companies. Now coming to this composition, I hope that everyone knows composition, but it consists of Suvarna Bhasma, Rajat Bhasma, Vang Bhasma, Naga Bhasma, and some herbs. And each of these components has been scientifically studied and scientifically reported to have anti-aging property, immunomodulator property, then cardiotonic property. Some constituents have also been reported to have excellent effect in obesity. So indirect scientific evidence of all these conditions suggests that VKR will be useful in diabetes-related complications. There are many papers published on this excellent formulation. Few papers we have came across that it is useful in management of retinopathy, type 1 retinopathy. Everyone knows that type 1 and type 2 diabetes, the complications, they may differ when it comes to pathological conditions. So this Vasanta Kusmakar Rasa has been reported to have effect in diabetic retinopathy. But as we know that 90% diabetic patients, they suffer from type 2 diabetes. Type 1 diabetes, of course, the patients, the number is considerable, but the majority diabetic patients, they suffer from type 2 diabetes. So we thought that this particular known formulation, it must be tested in type 2 diabetes related complications. So coming to this hypothesis or rationale for the present study, we know that unmet need related with treating diabetes and diabetic complication. There are no specific drugs approved by any regulatory agencies for managing, or we can say preventing, these diabetic complications which affect majority of diabetic people. Traditional classical formulation of Ayurveda focuses on holistic approach towards the treatment, and the main advantage of this is potency, effectiveness, and longevity. So that's why we proposed to study this particular formulation in animal models of diabetic complications. So these are the objectives. We studied this Vasanta Kusmakar Ras in type 2 diabetic nephropathy, type 2 diabetic neuropathy, cardiomyopathy, and type 2 diabetic retinopathy. Coming to the first complication, neuropathy, the increased prevalence of diabetes has inflicted the burden on human health, and patients suffering from diabetic neuropathy, they are large in number. And of course, when it comes to molecular mechanisms of this particular complication, there are very few theories which gives us a clear idea about what is happening at molecular level when it comes to diabetic neuropathy. Now, there are many pathways which are activated during these disease conditions. So, pathology of this disease is linked with expression of many proteins inside the cell or even at the nuclear level. And there is a sufficient literature. The science has moved ahead to explore all these mechanisms in detail. And as far as this diabetic neuropathy is concerned, we focused on one pathway, which is the major molecular pathway taking place inside the neurons, that is WNT beta-catenin pathway. Now this WNT beta-catenin pathway is expressed into the cells, leading to damage to the neurons. So we use this type 2 diabetes animal model, which consists of a low dose of streptozotocin, which is a toxic chemical for beta cells of pancreas, and high fat diet. So high fat diet along with streptozotocin, it causes type 2 diabetes, and if we will not give any type of treatment for the experimental animals, the animals will develop this type 2 diabetic neuropathy. So we gave treatment to all these animals for 16 weeks, and at the end of the experiment, we studied various biochemical parameters, then behavioral assessment, various inflammatory parameters, oxidative stress, histopathology, and we also studied expression of proteins at this neuron level, WNT and beta-catenin. Now, in each set of study, we took a standard drug. I told you that there is no specific drug for treatment of any complication. But the symptoms related with these diabetic complications, they are managed with the drugs which are already available in the market. So pregabalin, which is a known analgesic drug, we took for this particular study, and we used 2 doses of Vasanta Kusmakar Ras based on its clinical use. Now, coming to the results of this particular study, you can see that plasma glucose levels, they are decreased when we compare them with diabetic control animals. Now, these diabetic control animals will not give any type of treatment. The same thing happened in case of glycated hemoglobin, which is one of the important marker in case of diabetes. There was a significant reduction in the glycated hemoglobin levels in animals treated with this VKR at 2 dose levels. Even insulin levels, then HOMA-IR, oral glucose tolerance test, all these parameters, they showed protective effect of this Vasanta Kusmakar Ras in diabetic animals. I mean, even in case of lipid parameters, we got significant results where there was a significant decrease in triglycerides, total cholesterol, LDL, and increase in HDL levels in experimental animals. Now, behavioral parameters, they are designed for this animal model, which are having similarity with that of humans, that a patient suffering from diabetic neuropathy will be having changes in the sensation. It may be change in the sensation related with heat or pressure, etc. So we tested the thermal nociceptive threshold levels by using hot plate. Now, this is the first picture. You can see it's a hot plate where the plate is maintained at 55 degrees Celsius. So when animal is placed on the plate, it's a hot plate, so the animal will jump or animal will try to withdraw the paw from that hot surface. Now, this time is decreased in case of diabetic animals due to affecting the nerves which carries information to the brain related with change in the environment. In case of mechanical withdrawal threshold also, we could see that there was decrease in the response in case of diabetic animals, and we can see that there was significant increase in response time and the response pressure when the animals, they were treated with Vasanta Kusmakar Ras. So this indicated that this Vasanta Kusmakar Ras is having important function in protecting neuron, or it may be decreasing the progression of damage to the neuron in case of neuropathy. Now, we also tested nerve conduction velocity, which is also an important parameter when it comes to humans for diagnosing such type of condition. So, motor nerve conduction velocity and somatic nerve conduction velocity, both these velocities were recorded with the help of a data acquisition system. And we can see that this VKR treatment, it showed significant increase in the MNCV and SNCV when we compared it with diabetic control animals. So, as I mentioned, diabetic control animal, the motor nerve conduction velocity as well as somatosensory nerve conduction velocity will be decreased. All these pathies, they are associated with inflammation. So, all these inflammatory markers we could assess in the animals treated with Vasanta Kusmakar Ras or any standard drug, and we can see that there was significant decrease in majority of these inflammatory markers when they were treated with Vasanta Kusmakar Ras. We also assess this oxidative stress markers in neurons in this experiment. So, we know that when there is inflammation, it is linked with oxidative stress, and when there is oxidative stress, again the organ will be damaged at higher level. So, it is very important to manage this oxidative stress along with other mechanisms. And as the results, you can see that the oxidative stress markers like MDA, it was decreased in case of all the treatment groups, including the standard group. GSS, SOD, and catalase, we can see that the increasing trend in all these important antioxidant enzymes in the nervous tissue. Coming to histopathology, we could perform this histopathology of the sciatic nerve, which we isolated from all the experimental animals, and you can see that the damage to this sciatic nerve is less when the treatment group was examined for the histopathological changes. And at the end, as I mentioned, that WNT3 and beta-catenin pathway is the main molecular pathway in diabetic neuropathy, and we could assess the expression of WNT and beta-catenin in all these experimental animals. So it clearly says that WNT3 levels, they goes down after treatment with Vasanta Kusmakar Raj along with beta-catenin. So here we can say that this particular treatment, VKR treatment, improved this nerve conduction, velocity, and behavioral parameters, ameliorated the changes in plasma glucose level. It also improved the oxidative stress marker levels, and it prevented axonal damage, and it reduced the expression of WNT3A and beta-catenin. So from all these results, we can say that, we can conclude that this formulation, Vasanta Kusmakar Ras, has significant effects in diabetic neuropathy via inhibition of WNT3 beta-catenin pathway. Coming to the next complication, that is diabetic nephropathy. This is also one of the important complication of diabetes. Many a times it goes undiagnosed and the patient will suffer from chronic kidney disease or even end-stage kidney disease. So it's typically associated with arterial hypertension and increased cardiovascular mortality and morbidity. Now coming to this pathway, the molecular level pathway in diabetic nephropathy, TGF-beta, transforming growth factor beta 1, and SMAD, these two proteins, they are overexpressed in kidney in case of diabetic nephropathy. So we targeted this particular pathway, and we proposed that targeting this pathway, inhibiting this pathway will decrease the progression of diabetic nephropathy. The protocol was same. All the animals, they were administered with septozotocin and high-fat diet, and the treatment was given for 16 weeks. But the endpoint parameters were slightly different. We could able to study various urine parameters. Then different types of stainings were used to assess the changes in the histological structure of kidney, and inflammatory marker and oxidative stress also we could study. Coming to this kidney function test, as everyone knows that plasma protein, urine protein, this ratio is very important. There was a decrease in the plasma protein levels of diabetic animals, and the levels were increased after treatment with Vasanta Kusmakar Ras. Albumin, which is one of the important protein, the same trend we could observe, and plasma creatinine and blood urea nitrogen levels were also decreased after treatment with Vasant Kusmakar Ras for 16 weeks. Now the important thing is that this is a type 2 diabetes study, so that's why the long-term treatment was selected. 16 weeks each animal was treated with a specific dose of VKR. And we know that it's not a chronic condition, it's not an acute condition, but it's a chronic condition which will require a long-term treatment. Now urine parameters also, we could get significant results like GFR, glomerular filtration rate, which is severely decreased in case of these animals suffering from diabetic nephropathy, and there was increase in glomerular filtration rate after treatment with VKR at 2 selected dose levels. Again, the inflammatory marker levels were also increased in all diabetic animals, and there was significant decrease in TNF-alpha, IL-6 and IL-1 beta, which are the specific markers in inflammation. There was a significant decrease. Antioxidant level enzymes, again same trend as that of neuropathy. We could see that all these antioxidant enzymes, they were increased in the specific tissue, that is kidney tissue. Now we studied structure of kidney at the cellular level by using different types of staining like HE staining, which is regularly used staining in majority of histopathological studies. So you can see that there is increase in glomerular thickening when it comes to HE staining, and the glomerular thickening was decreased at the highest dose of VKR. So it says that the VKR is protecting all these tissues at specific level. Coming to this periodic acid staining, it is this is a special type of staining which is used for understanding the expression of glycoproteins. So abnormal protein, and due to abnormal protein synthesis, there is thickening of specific area like glomerular basement membrane thickening. It can be easily observed by using such type of staining. That is, we call it as PAS staining. And Masson's trichome staining, it is specific for fibrin, which is important protein in fibrosis. And as we can see here, the fibrosis, the blue color in the slide, it indicates the increase in fibrosis. But if you will see the picture of kidney, VKR treatment at highest dose, 56 milligram per kg, you can see that very small amount of fibrin has been deposited at tissue level, which indicates that there is decrease in renal fibrosis. We also studied TGF-beta1 and SMAD in the kidney of all these experimental animals, and there was a significant decrease in this TGF-beta levels at both these doses of VKR, and even the SMAD 2/3 expression was decreased in all these tissues. So even you can just go through this diagram where, lisinopril was used as a standard drug in this study. Again, everyone knows that lisinopril is not an anti-diabetic drug, but it is an anti-hypertensive drug, but it is approved for its use in diabetic nephropathy. So we took this lisinopril as a standard drug, but as the results suggest that even the lisinopril is not significantly reduced this TGF-beta levels in the kidney. So at the end, we can say that VKR has significant effects in diabetic nephropathy by reducing expression of these two important proteins, TGF-beta1 and SMAD2. Coming to cardiomyopathy, it's a macrovascular complication of diabetes, and here we specifically took into consideration PI3K and AKT signaling pathway in cardiomyopathy. We also studied the role of GLUT4. GLUT4 is nothing but what? It's a channel protein which helps glucose entry into the cell. So obviously we'll be requiring more number of GLUT4 at cellular level to reduce the blood glucose level. And in diabetic patients or in diabetes, the GLUT4 levels, they goes down. The cell cannot synthesize GLUT4 at higher rate. Animal model was same, the treatment pattern was same, but there was slight differences in the parameters which we selected for this cardiomyopathy. Coming to this specific hemodynamic parameters, all heart-related parameters were studied by using data acquisition system. We could ligate this particular blood vessel so that we can record all these parameters by using data acquisition system. So heart rate, which was decreased in diabetic animals, it was significantly increased in the treatment animals at both the dose levels. Even PR interval, QRS interval, R wave amplitude, then systolic blood pressure, arterial blood pressure, all these parameters, they were improved after treatment with VKR for 16 weeks in diabetic animals. Now these are representative ECG graphs of experimental animals, that is Sprague-Dawley rats. Now coming to cardiac markers, CKMB and this troponin, these are two important markers which indicates damage to the heart or inflammation to the heart. And this CKMB and this troponin levels, they were increased in case of diabetic animals, the animals suffering from cardiomyopathy, and these levels were significantly decreased after treatment of this Vasanta Kusmakar Rus to all these animals. Again, similar pattern we could observe for oxidative stress in heart. Even in histopathology, we could see that there is less damage at the tissue level in different types of staining. It may be HE staining, Masson trichome staining, even the fibrosis was decreased in diabetic animals treated with VKR. And in case of protein expression study, this PI3K, AKT, and GLUT4. You can see that all these important proteins, the levels, they go down in case of diabetic condition. And this VKR treatment, it showed significant improvement in the expression of these proteins which are important for the cell. So yes, this Vasant Kusmakar Ras showed significant effects in management of diabetic cardiomyopathy by activating this PI3K, AKT, GLUT4 pathway. Coming to this last microvascular complication, that is diabetic retinopathy. Again, there is no drug for management or treatment of diabetic retinopathy. The best way is to retard, to inhibit progression of all these complications by controlling different pathways. So here we took VEGF, vascular endothelial growth factor, and matrix metalloproteinase, that is VEGF MMP signaling pathway in retinopathy. And polyol pathway is also very important, which is related with this diabetic retinopathy, one of the important microvascular complication of diabetes. Now here the model is slightly different. As you can see, the rats, they were treated with streptozotocin, but 8 and 1, that is 9 weeks, they were kept as such. And after 9 weeks, these rats were treated with VKR and calcium dobicelate, which is a recently approved drug for management of diabetic retinopathy. It's not a specific drug, again, just a symptomatic treatment. 16 weeks treatment was given to each animals. So this was the, what you can say, longest animal model we used for studying this diabetic complication, that is diabetic retinopathy. And we could record electroretinograms of all these experimental animals. We did fundoscopy, biochemical parameters, histopathology, and studying the expression of VEGF and MMPs. Coming to this fundoscopy, as we can see that there are diabetic eye, it shows presence of newly developed blood vessels, angiogenesis, and it's of course a marker in case of cancer also, but angiogenesis is typical in case of diabetic retinopathy also. So you can see the number of blood vessels, the increased number of blood vessels in the diabetic control animals. And the animals which are treated with calcium dobicelate and Vasant Kusmakar Ras at 2 dose levels, they show the less angiogenesis, which is very much required for controlling this type of complication. Then histopathology of retina, the hallmark of this condition is thickening of retinal tissue. And at the end, there is decreased in a function of retina, which is very important for judging different types of colors through the visual pathway. So there was a significant decrease in the retinal thickness in all these treatment groups. Then we recorded electroretinogram of all these experimental animals. Now electroretinogram is nothing but what? Just like ECG, we study the pattern of stimulus and interpreting that particular pattern by using some software. So ERG study was carried out by using this data acquisition system. So you can see this setup of ERG study. The rat is in that chamber and the lights, they are switched on and switched off. So when the lights are switched on and switched off, the action potentially generated into these retinal cells and which is processed in the brain. So this particular processing of these actions is decreased in case of diabetic retinopathy. And we could record all these changes in eye at different light intensities. And all these recordings at different light intensities showed that there was a significant effect of this VKR when the animals, they were treated for a long time. So these are important parameters like A wave and B wave, and we could record ERG of each eye, that is left eye or right eye. So we, generally in case of animal experimentation, we keep one eye untreated and we just go for recording of all these ERGs in one eye. But we could able to record all these ERGs in both eyes in each animal. So this is the ERG pattern at 50 lux. So we can see that this A wave changes are prominent in case of diabetic animals, and the A wave pattern is increased in case of the treatment animals. So this is the data for 100 lux, this is data for 316 lux, and all these ERGs, they could clearly presents the improvement in the structure of eye or decreasing the progression of damage to the eye. Then we studied these vascular endothelial growth factor and matrix metalloproteinase 2 and 9 in eye tissue, and you can easily make out that all these proteins, they are decreased in case of diabetic animals, and after treatment there was significant increase in these proteins which are there in eye of all these experimental animals. So from this study we can conclude that this Vasanta Kusmakar Ras has significant effects in diabetic retinopathy by suppressing this MMP2, 9 and VEGF proteins and improving the electrical conductivity of light into the eye. Now this work, we did this work to support this use of Vasanta Kusmakar Ras in preventing, or you can say prolonging, these diabetic conditions. So the present study work provides sufficient preclinical evidence that VKR treatment has holistic approach towards delaying the progression of diabetic complication, and the study provides a scientific support for framing a proof of concept of usefulness of this classical Ayurveda formulation in diabetes and associated complications. So this molecular level study will give scientific evidence for using Vasanta Kusmakar Ras for preventing diabetic complication in patients. Now this is my research team, all are my PhD students, and the majority of them, they have worked on various aspects in diabetic complications. Thank you everyone.